Hi everyone, this is the final video for this series on medulloblastoma meta-analysis portal. I work as a computational scientist at St. Jude and my role goes further into project management, strategy, and communications, where I help my team understand the user's perspective from a biological standpoint, as well as managing the testing and validations of the pipelines to make sure data management is seamless. Now, this involves active communication with internal and external stakeholders such as oncologists and statisticians, so we can provide rigorous analytics and visualization solutions for such complex data. So in this video, I will show you an actual biological use case so you can better understand the disease context and how this portal can act as a bridge for investigating data discrepancy issues as well as a novel discovery platform. So as I showed you in the previous videos, this MB Meta portal comprises data on a multivariable level from 898 patients that were enrolled in 3 large clinical trials. And we looked at how to analyze certain types of visualizations, such as summary plots, Kaplan-Meier, Cox regression, lollipop plot, the methylome TSNE, individual sample level data using sample view, and then also looking at cases across the cohort using the sample matrix. Today, we will look at some biological use cases that I have found using this portal. But before that, let's talk a little about what medulloblastoma is. Now, very early on, when medulloblastomas were identified back in the 1920s, they were grouped together with the other primitive neuroectodermal tumors. There was no clear strategy on how to treat these tumors because of a lack of a clear classification system. Now, medulloblastoma itself is a very rare and the most malignant brain tumor in children. It's only second to leukemias and it's highly heterogeneous, and that's why the classification is still not there yet. It's still a problem, and it's still a gap in the field. So this brain tumor starts in the lower back part of the brain called the cerebellum, as you can see in this image here, which is involved in muscle coordination, balance, and movement. Now, it's classified as an embryonal tumor by the WHO because this is also a tumor that can metastasize through the nervous system, taking advantage of the cerebrospinal fluid. And the convention accepted by WHO is based on the location or the origin of the tumor amongst other criteria. But recently, a lot of molecular studies have been coming out, and those provide a better insight into the classifications of these tumors. So, since 2016, WHO has adapted and started considering both not just the phenotypic but also the genotypic characteristics for these tumors. Now, the latest is the 2021 classification, which is shown here, which classifies medulloblastoma, into two different ways. One is molecularly defined, and one is histologically defined. So, molecularly defined, you have the medulloblastomas, which are Wnt activated. Then you have medulloblastoma, which are sonic hedgehog activated, but TP53 wild type. You have medulloblastomas, which are sonic hedgehog activated and TP53 altered. And then medulloblastoma, which are non-Wnt and non-sonic hedgehog, which is an entire group on its own, and we will come to that. So, what is TP53? It's the guardian of the genome involved in DNA repair and triggers cell death if the DNA is damaged. So, mutations in TP53 alone are actually very deleterious to the cell. This is, basic biology 101. Um, the other classification, which is histologically defined, segregates medulloblastomas into desmoplastic nodular medulloblastoma. These are more fibrous with nodules embedded between those fibrous tissues. And then there's medulloblastoma with extensive nodularity. So there's an extensive formation of nodules in this. Then the large cell medulloblastoma, which are large round cells, and this aggressive form tends to grow and spread even more rapidly. And then you have the anaplastic medulloblastoma, which appear very abnormal and very aggressive under the microscope. So let's go on to look at these different groups within medulloblastoma, which are molecularly defined. Now there are 4 major groups, and some of these groups are further subdivided into subgroups, and we'll look at both of them. So just remember there are 4 major groups, and then some of these groups are subdivided even further. So the first one is this Wnt, which is shown in the blue. group which is based on the activation of Wnt signaling and constitutes about 10 to 15% of the cases. One of the major mutations that is prevalent in this is beta-catenin mutations, and most of them are missense mutations. And this was an example with CTNNB1, which I have showed you in the previous video, so you can always go back and look at that. Now, this particular group occurs in children older than 4 years and is often found in one area of the brain when diagnosed. So if you look at the anatomical location, ends up infiltrating the brainstem. The chances of surviving 5 years after diagnosis is over 95% for children and about 80 to 95% for adults. One of the interesting things about this molecular group is the presence of a leaky blood-brain barrier. So there can be hemorrhagic bleeding that can occur as a result. And because of having this leaky blood-brain barrier, they're usually, treatable with just chemotherapy. So which is why their prognosis is pretty good generally. The next one is the sonic hedgehog, which is in red. And these account for about 30% of all the medulloblastoma. These tumors are also found in infants under 4 years old and adults over 16 years old. So, either end of that spectrum. But they can also occur in children in between that age group. These are characterized by mutations in genes involved within the sonic hedgehog pathway, such as patch1, SUFU, or SMO. And now this group is further subdivided on TP53 mutation status. So Wnt was not subdivided further, but this one is subdivided further into TP53 mutation status. So the ones that are wild type are more common and have a better prognosis, but the ones that are mutated for TP53 are more aggressive and have a poorer prognosis. And their histology can be different, and there can be variations in their histology. Now, where do these occur? So these sonic hedgehog tumors occur in either of the cerebral hemispheres, especially in adults. And their prognosis is dependent on the TP53 status and other mutations, but generally they have an intermediate prognosis. Now, the last one, the group 3 and group 4, there are the non-Wnt and the non-sonic hedgehog. They're generally combined together, and together they account about 60% of the all medulloblastomas, with group 4 being the most common. Now, the reason why they're combined together is because they have overlapping molecular features, and they are not associated with any specific pathway, which makes the demarcation between these two groups very difficult and very ambiguous. So these tumors can occur at any age but are mostly common in children. And since they are heterogeneous, owing to the fact that there is a lack of signaling pathway that is driving these tumors, there are different molecular alterations that you can find overlapping, and one of those is MYC amplifications. Now, what is MYC? MYC is a proto-oncogene, and that plays a crucial role in cell cycle regulation. So it encodes this transcription factor that can go and bind to the DNA and activate the expression of genes involved in cell growth and proliferation. So if you imagine, like, there are more copies of this gene, then basically there's more growth factors that can lead to a higher aggressive growth in the cell. So because it's a proto-oncogene, activation or amplification of MYC alone can cause havoc in the cell. The other frequently shown alteration is the isochromosome 17q, and that is mostly seen as group 4, and we'll look at some of this data on the portal. As compared to group 3, group 4 has more of these mutations, but they seem to have a better prognosis. Now, both of these groups can exhibit, the large cell or the anaplastic, which are the more aggressive histologies. And because they're associated with MYC amplifications, they are more aggressive in nature. Where do they occur? They occur in the midline, as you can see. And these are further subdivided into 8 subgroups across group 3 and group 4, which we will talk about in the coming slide. So, like I said, the demarcation between group 3 and group 4 because of lack of a unifying molecular signaling pathway is very poor. And that is shown in this 2017 study where this group published a landscape study which analyzed 491 sequenced medulloblastoma samples and around 1200 epigenetically analyzed cases. So, for their epigenetic profiling, they used DNA methylation to classify these tumors into different subgroups to understand the epigenetic landscape of these tumor samples. So basically, a machine learning method for dimensionality reduction is applied to the methylation array data, and then that generates this clustering pattern on a 2D scatter plot. And then supervised machine learning can be performed for annotating these samples on the clusters. So in this TSNE on the left, you can see that there's a distribution or clustering of tumor samples according to the methylation data. And then on top is overlaid the subgrouping for group 3, group 4 tumors, which distributes them into several subgroups that those four main subgroups are stratified across. So you can see that with subgroups within group 3 and group 4 are not well separated and there is some overlap between them. So this TSNE is only showing um, the TSNE and the matrix plot on the right are only showing group 3 and group 4 samples. We are not discussing the Wnt and sonic hedgehog just for this video. That was just an introduction. So you can see that there are these black curves that are drawn over this scatter plot or the TSNE to be able to distribute or divide these samples, but they're not very there's not clear boundaries. So you can see that there are some samples that kind of cross over into another cluster, or there are some sort of overlapping. And we can also see that in the molecular features as reported in this matrix. So what they have done is that they have also found these subgroup specific driver mutations, and these basically become, new actionable targets which are based on these epigenetic studies. Now, if you want to know more about epigenetics, check out my latest blog post that I wrote with GSA. The link is in the description below. So in this matrix, we can see that MYC amplifications occur in subgroup 2, 3, 5, and 7. And then you can also see KBTBD4 indels, which are insertions or deletions that occur in subgroup 2 and subgroup 7. So this basically tells us that, this group is very heterogeneous. Some of these mutations are overlapping across the different subtypes. And so the classification is still not very clear. So let's get back to the portal now, and let's look at some of these use cases and examples from what we just talked about regarding the different molecular groups and different molecular subgroups within those groups. And here I have built this pre-built matrix. I have removed some of the variables just to make it simpler to understand. And so you will see that each row within this matrix visualization is a type of variable or is a type of category. And you can reorder and move things around based on how you want to sort some of these variables. So now each row is a variable containing information about that variable or that category, and then each column is a sample. So on the top here, you will see some of the demographic or the clinical variables, such as the molecular group, the molecular subgroup, the metastasis status, and then the CSI group. Also, the biological sex, age at diagnosis. Now, age at diagnosis is both shown as a discrete or a categorical term, as well as a continuous. So you can see some of the histogram related data or the continuous distribution for that age, for those age groups. And I have further subdivided this matrix into the main molecular groups, the four main molecular groups, which is your Wnt, sonic hedgehog, group 3, and group 4. And when you hover over them, it gives you the number of samples and also the number of samples within each of these distributions. So here you can see within somatic mutations, the number of samples that fall into the different types of mutations. Same goes for the germline mutations, copy number variants, whether it's wild type, whether it has a gain for that gene, or whether it has a loss. And then all the other clinical variables also show up with their numbers in the parentheses. And so based on this grouping, the underlying biology is something that you can visualize and look at and draw trends from. So you can compare whether something stands as an outlier or there is something interesting to note which has not been previously shown or published. So one example I can give you is CTNNB1, which is beta-catenin, which we also talked about in the previous video. So you can always go back to those previous videos. Only shows up in the Wnt group because it is a classic feature of the Wnt group, and you won't see it in any other group, and that is expected. Then if you look at MYC amplifications, now MYC amplifications, as we talked about, are associated with poor prognosis, and they are supposedly a feature of the group 3 medulloblastoma. But you will also notice that within this cohort, there are a few cases which also fall into group 4. Again, reiterating the fact that these groups are not delineated really well. There's not enough demarcation. The other example to really observe that is also the KPTBD4 mutations. As you can see, these are protein insertions. That means there are sequences of amino acids that are inserted in specific regions of the protein domains. And we'll look at that in more details in the next few minutes. So you can see that KPTBD4 here is present in both group 3 and group 4. And another one I want to highlight is the status of the isochromosome 17. What that means is that there is a loss of chromosome 17p arm and there is a gain of chromosome 17q arm as assessed by FISH or in situ hybridization. And generally, these are also associated with a poor prognosis and are a feature of group 4. But you can see that there are cases that, or in within group 3, that also show this chromosome iso 17 feature. And so the question really becomes that since MYC and both chromosome iso 17 are poor prognostic markers of group 3 and group 4, which one really takes a precedence over the other? So the way to assess that is using survival outcomes as an outcome variable and then assessing which of MYC or chromosome iso 17 has a worse impact on survival and how do those both tie in together within this cohort. So let's look at the survival outcome analysis. For that, I am going to close this matrix. So to assess the impact of MYC amplifications versus ISO 17 on survival outcomes, we can create groups based on mutual inclusivity versus exclusivity and using both of these factors. So to do that, we will first, filter for group 3 and group 4 because MYC amplifications as well as your isochromosome 17 are restricted to this group of tumors. And then the other thing we also want to filter down is for the average or the standard risk patients, because the ones that received low dose craniospinal irradiation are more experimental. So those that received low doses and also carboplatin treatment are more experimental because this SGMBO3 trial is trying to reduce the dosages in medulloblastoma patients to show that they can achieve the same treatment success as the high risk groups, uh, even reducing the dosages, because with higher dosages, you see a lot of adverse events or side effects in survivors with medulloblastoma. So let's go ahead and also remove those cases. So I'm going to type carboplatin with CSI, and I don't want those cases like I just mentioned. And then also the CSI group, I just want standard dose and high dose. So you can see that there's 458 samples. And now using this subcohort, we are going to create groups which will include in ISO 17 or MYC, and we'll do certain combinations of those. So the first thing I would want to do is create the group for isochromosome 17 because we're trying to assess whether MYC amplifications have a worse prognosis on survival outcomes for these patients, or it is isochromosome 17. Which one takes precedence is the question. So I'm going to type in 17p for 17p loss, because that is what your isochromosome 17 is, and then 17q gain. And this is the default is shown in green color, and you can change that if you like. I'm going to call this group ISO 17. There are 212 samples in here, and I'm going to go ahead and create this variable. So now there is this button that shows up in blue, and you can click on it to do extra analysis. But I'm going to go ahead and create my other groups using this variable now. So I will add the group and go to mutation and type in make. Now what we can do is we can choose make wild type cases and ISO 17. So now what I'm making sure is, so the first group that I created with ISO 17 was regardless of the status of make amplifications. So it doesn't matter whether make is amplified or not in that group. But in the second group, I'm specifically choosing cases. So you can see now there are 203 samples that have ISO 17, but they do not have make amplification. So they're wild type for make. So I'm going to call this ISO 17 and no make, basically. And then I can create, I can create that as my second variable by deselecting the first one. So now I have ISO 17 and no make versus others, right? Now I want to add in a third group, again using make. So now I will choose make with copy number gain. So now I want make amplifications in these samples. So there's 20 samples that have make amplifications, but I also want ISO 17. So now that's 9 samples. So there are 9 samples that have make amplifications and as well as have overlapping ISO 17 mutation chromosomal status. So ISO 17 and make amplification or make gains. Let's call it make gains. It's shorter and easier. So deselect 1, 2, and then create a group. And now, what do you think the last group should be? So the last group should be only make. Now we want to see cases with make. So those would be those 29 cases. And I will still add ISO 17 versus others, but I'm going to negate that. So there's 11 samples that can only contain make amplifications, and they're not present in the other group. So I'm going to call this make only, and then deselect the first 3. And then I'm going to create a group with make only. So you see what we did? We're creating combinations where we're including ISO 17 and excluding make, and then the other cases where we're including both of them or excluding ISO 17 and keeping make, right? So this sort of a analysis will tell us which factor or which mutation or which, chromosomal aberration really has a huge impact on survival outcomes. And for this, we're going to use the progression-free survival because that gives a real assessment of how the treatment, how successful the treatment was. Because your progression survival, if we go ahead and look at the definition right here, is the time interval from the date on study to the date of the progressive or relapse of the disease. So basically, it's accounting for the progression of the disease or the relapse of the disease after the treatment, right? So whether the disease has actually progressed or not. So it gives a really good measure of the success of the treatment regimen. Now we have all our groups created. So there's ISO 17 versus others. So that contains everything regardless of the make amplification status. And then you have ISO 17 and no make versus others. So now this contains your ISO 17 without make amplifications. And then the third group is your ISO 17 with amplifications for make versus others. And so now we are accounting for make in that. And then the fourth group is only make gains and there is no ISO 17. So let's look at their survival outcomes one by one. So I'm going to go and click on progression-free survival. And what do we see here? So the green one is the ISO 17. Now remember, this is the group that also contains make cases. And when you look at the rest of the group 3, group 4 cohort, so we have filtered for those, right? We are looking at only group 3, group 4. So the others are all the rest of the cases that do not fall into this ISO 17, that do not have ISO 17 chromosomal aberration. So you can see that the p-value is not significant. That means there's not a difference in the survival outcome when cases have ISO 17 chromosomal aberrations, when there's a loss of the P arm and a gain of a Q arm with chromosome 17 versus the rest of the cases. So their survival outcomes over this time period, from 0 to 10 years, is pretty much similar. That's what this tells us. So now let's go and open the other one. So now we're going to look at only ISO 17 without MIC. So only ISO 17 without MIC. Again, the cases with ISO 17 that do not have any MIC amplifications also do not show a difference in the survival outcomes from the rest of the subcohort from group 3 and group 4. And you can see that really nicely here in the Kaplan-Meier curves. And then the third one, I'm going to open that. Right. So here is where things start getting different, because now we're looking at ISO 17 and specifically only gains for MIC, right? So we've selected MIC gains here. So we know that the cases that harbor ISO 17 chromosomal status also have MIC amplifications. And you can see that those cases have a very poor progression-free survival. You can see the immediate drop for those cases compared to the rest of the subcohort. And this is very significant as shown by the p-value. So this clearly tells us that between ISO 17 and MIC, when these cases have MIC amplifications, their survival outcomes become even poorer. And that's what we have seen from the previous analysis as well. So to confirm that, we will only look at now the MIC amplification. So the cases that only have MIC amplifications. And see, here again, the cases that don't have ISO 17 but do have a MIC amplification, that only have MIC amplifications, follow a similar curve as similar to the previous plot, which did have ISO 17. So what this is suggestive of, that MIC is the major prognostic factor for progression-free survival to assess the progression of the disease for this subcohort of group 3 and group 4 within the standard and average risk patients. And ISO 17 itself is not playing a major role as a prognostic factor, even though that has been published in the literature for other studies. So, but for this particular data set, we can clearly see that these cases that received low dose and high dose CSI and did not receive carboplatin treatment, MIC was the major deterministic factor in their progression-free survival. And this is how you can set up some of the complex survival analysis and look at Kaplan-Meier curves. And the legend also provides you the number of the patients that were lost to follow up or that were censored. So what we saw was MIC with ISO 17, and some of these are very specific to specific groups or specific subtypes. So what this group has shown are that there are driver events that are very subgroup specific that are driving the disease phenotype. And one of them we saw is MIC. The other one that they have reported is KBTBD4. What is this KBTBD4? This KBTBD4 is a adapter protein that assists culin-3 mediated ubiquitin-dependent degradation of small proteins or cofactors. What that means is that it provides a platform for degradation of small cofactors, which could be co-repressors that are responsible for repressing certain parts of the chromatin. And how it does that is through its protein domain, which contains these Kelch repeats. So the one that is shown in lavender color here, as you can see. And there are conserved regions which are conserved from species to species within the Kelch repeat, which we will see in a little bit. And there are insertions of amino acids, or there are missense mutations as shown here in this lollipop in figure A. And because of having these insertions, what ends up happening is the loop in which those substrate binding pocket exists extends to allow for maybe new or novel substrates that can bind there, which are not supposed to get degraded, by the way, but end up getting degraded as a result in the disease context. And so what they have shown in this is that these KBTBD4 insertion mutations are very specific to certain subgroups, like subgroup 2 versus subgroup 7. So on the methylome T-SNE, you can see that it forms two distinct clusters. And this is very interesting because subgroup 2 has a very poor survival when compared to subgroup 7. Subgroup 7 seems to have a much better survival. And we did run some tests on the portal within this MB-Meta cohort to see if KBTPD4 is attributing to those survival outcomes. And the sample size becomes too small to make any solid conclusion. So far, we think that it's probably not the factor that is driving survival outcomes in this cohort, but it is definitely driving the disease phenotype. And how is it doing that? A study in 2022 published this paper where they artificially produced these mutations which were found in clinical studies. So in the Kelch2 repeat, and we'll look at this the sequence of this Kelch repeat in a little more details. In this Kelch2 repeat, what they did was they created mutations that are clinically reported, by the way, at P311 pp. So they introduced two prolines, and then at R313 they introduced proline and two arginines. So what ends up happening is, as you can see in this colorful diagram below it, where there is the loop of the KBTPD4 Kelch2 repeat shown, you can see that there is an increased length of the loop, and that is where there is the substrate binding pocket. And what happens is, if that loop is extended, then there's a possibility that something slightly bigger than the normal residue that fits into the wild type pocket has the capability of fitting into this pocket and getting degraded. And that is exactly what they found in their immunoprecipitation and mass spectrometry studies. They found this small molecule called CoREST that can bind into this pocket and get degraded. But why is that important? So CoREST, along with histone deacetylase and LSD1, forms this repressor complex that will bind active chromatin and repress gene expression during normal cerebellum development. And that's really important. But what happens in the disease context when KBTPD4 is mutated, right? When there are insertions in that loop, CoREST can fit into that pocket. Now it's a substrate for KBTPD4. It can get ubiquitinated and as a result get degraded, which is not supposed to happen. But in the disease context, that is what happens. And as a result, you have an active chromatin signature. And the genes are getting expressed now. The genes that are getting expressed as a result are stem cell factors that are pushing the cells or the tissue in a more mesenchymal or in a more stem cell-like signature. And that's why this is how it acts as a driver event. So in this cohort, like the reason why we encountered this was on the TSNE, we also noticed that within the 898 samples, yes, there are the two clusters for your subtype 2 versus subtype 7, but we additionally saw other insertions and mutations within subtype 1, subtype 2, subtype 8, as you can see in the TSNE. And this is an old figure or an older visualization of the TSNE. Because what I noticed was there was this nonsense mutation within KBTPD4 hiding, very tightly clustered with the subtype 2, subgroup 2 mutations. And that got me thinking, because if we go back and look at the structure, right? So this particular, here, let me zoom that a little bit. And right. So this PRRMWKCNN, this particular structure, this particular amino acid sequence is conserved from species to species. And that's the one where substrate binding specificity takes place. The substrate would interact with these amino acids. And that is what I was showing you from the study in 2022, where they inserted an extra PRR at this R313. So this R is 313 and this M is 314. This particular isoform is slightly different, which is why like the numbering is different. So don't pay attention to that. But what we noticed was this M seems to be a nonsense, right, at M314. What that means is, for a biologist, that would mean that since that's where a truncation takes place, none of these amino acids that are after that M314 would get translated into a protein. That would leave some sort of a gaping hole in this particular Kelch repeat. So it would be completely, all the amino acids before that would be completely exposed. And which doesn't really make sense, because biologically, if you think about it, such a protein should not be even existing in the cell, right? And so that is what something got us thinking as to what is really happening here? Is this even correct? Because these mutations have been called using the standard variant calling pipeline like GATK, for example. And so the next question was really, are these accurately called? Because normal standard variant calling pipeline, this becomes really difficult to catch because these appear as normal sequences. They're not really considered as mutations when you compare it to the reference. So that's the reason why sometimes such complex indels, which are insertions or deletions, can be difficult to catch. So let's go back to the portal and really see how to visualize this. So I have already opened this and I can just redo this again. So again, going back to charts and then selecting a scatter plot, which is your methylome piece name. Now, instead of coloring by molecular group, what we're going to do is we're going to color it by the mutation, which is KBTPD4 itself. And then once we have that, we can zoom in to see. What I'm going to do is hide the CNV wild type, also the germline mutations, and also the not tested. Okay, so now there's a lot more wild type in the background that you can see because these are somatic insertions or deletions. That means that they have occurred during the lifetime of the individual, and these are not inherited. So they're not germline and they're not copy number changes, which is why I hid all those other categories. So now you can see that there are these two distinct clusters on top, but let's also go ahead and put the shape using molecular subgroups so we can see what different subgroups they belong to. And now you can see that on top are your subgroup 7 and below are your subgroup 2. And the mutation that we were thinking was a nonsense is actually turned out to be R313 with the insertion of a proline and two arginines. So this is this one that is similar to what has been reported clinically before in that 2022 paper where they showed that CoREST is a co-repressor that binds and gets degraded that I just talked about. So we additionally found other complex insertion events. So one of them is this TSYKL, which is your threonine, serine, tyrosine, lysine, and leucine at I310 position. And the other complex mutation we also found was R312 with a proline, threonine, serine, isoleucine, histidine, methionine, and leucine. So this is like very interesting that because you notice that most of the subtype 7 are single codon insertions. So there's only single amino acids that are inserted, except for this one, which is multiple codon insertions. And then what we also noticed what within subtype 2, there are subtype 7 carrying insertions, which are multiple codons, like this PRR and the TSYKL, which is actually subtype 7, but it is sitting very close to the subtype 2 cluster. Additionally, we also notice subtype 3 containing PRR. There are two cases that are overlapping and sitting very close to this subtype 2 cluster. And then we also see frame shifts for subtype 8. We see another subtype 2, which is sitting away from its remaining of the clusters. And then a mutation in subtype 1, which is a single codon with the proline. So the question naturally becomes that, are these potentially misclassified? And that's what we are trying to show here, that we need more sensitive classification techniques, and that is a gap in the field because we don't know enough about the biology of these tumors. And because we don't know enough of the biology of these tumors, and we need more sensitive classification methods, can we use some of these factors to inform decisions in the clinic downstream? Because at the end of the day, the patient is risk stratified based on several factors, which not only include clinical factors, but also genomics factors. And then putting all of this together in a multivariable fashion is what would bring a change in the treatment and also the quality of life of the patient that is receiving the treatment. And that's where this portal becomes really, really important, serving as a novel discovery tool that is bridging the gap from your biological fundamental research into practices in the clinic where decisions are being made on how to treat the patient. And so some of these things we have talked about in the paper. And so once the paper is out, you can go and look at it. But the question is also, how did we look at these mutations and how did we correct some of these mutations and how did we find some of these mutations. So we have our in-house tool called as the ProteinPaint BAM track, and that is what I used to manually verify some of these mutation calls that were called initially by the variant calling pipeline like GATK. And so how we have used this is you have your reference genome, and I have just zoomed into the region where this particular conserved repeat in the Kelch2 domain is present. And there are certain parameters that you have to use. Now, these are BAM files directly obtained from the raw sequencing data. And what I've done is I've just positioned or aligned the sequence of these BAM files in accordance with the reference genome. And that's how I was able to look at some of these mutations. So this one, for example, at chromosome 11, at this position 4757360, there are 42 reads supporting the alternate allele. So within the URL parameters is what I had to add the variant itself. So if I go down this URL parameter, you can see that I had to add the variant in here. And so manually, I had to correct for this variant, and that's how it's able to now map 42 reads to the alternative allele. So I, as a user, have to provide that alternative allele for this. And it's more like a trial and experiment. So if you don't know what the alternative allele is, it is going to show you these blue colored digits, as you can see on top of the sequence that is aligned. And if you click on that, it will tell you what that alternative allele could be. And this is what you can copy and paste as part of the URL parameter right here where it says BAM and variant. And so you add that variant as part of the parameter and then reload the chart, and then it will show you how many reads are supporting that alternative allele versus how many are not. And that's how you get a sense of the correct number or the correct pattern of this alternative allele or the variant. And after you have corrected the variant, you can again rerun your annotation pipeline. We use the VEP, the ensemble VEP, and that is what was used to correct these variants. Now, all the details, the methodology, and everything else is provided in the paper itself. And once it's published, I will add the link in the description below. And that's how you can utilize some of the tools that are housed within ProteinPaint to make some of these discoveries. Keep an eye out for the paper and you will get a lot more information from the article itself. It's a very complex study and the paper itself is also quite complex to understand, but biology is complex. For me, I embrace that complexity of life.